2015, again, this time around, there's a lot of papers that actually talk about looking at what do they have now after about 7-8 years of NGS. This is paper number 28. Yes, finding variation is one step, understanding them is another. So they've been another progression step. They've realized that, yeah, you have all this, you have association, but what does it mean, right? So at the moment, people are more keen to understand, okay, I've seen all this association, so what can it do? What does it do? Understanding that is another. Functional characterization and mechanistic elucidation of the NSPs and genes actions are the next major challenge. So, I mean, this is where I would actually recommend whoever is interested in research to venture into bioinformatics, because there's a lot of data. There's a lot of data. No, there was a lecturer in bioinformatics. He's now left. He has joined Mayo Clinic. What he does every day, he doesn't need PhD students. He doesn't need a lot of research money. What he does is sit down, because you see him, when people do all these genomic studies and do all this NGS, there is now, if I'm not sure whether it's a requirement or not, but there's a database that you can deposit your sequences. So what he does is that, okay, let's take all, because they are like sort of available for public. So he downloaded them and says, okay, this person did heart study, this person did whatever related, and then they try to see. And his job is what he does is to summarize all these different studies using bioinformatics and then present it in a visual. There's another paper on visualization. Visual meaning, you know, when you see mathematics, it doesn't really mean much to you. But once you present it on a graph, then probably it means more, right? So you can understand more. So this is what it means by visualization of data. So that's what he does, and he's good at it. So he gets paper in very, very reputable journal. But he's no longer with us. He left for Mayo Clinic. Yeah. So yes, so now people realize, yes, there's a lot of variation, there's a lot of data, but what does it all mean? You need to start connecting the dots. Paper number 29, also another review, the variation game, cracking complex genetics disorders with NGS and omics data. Plus now, it's not just genomics. You've got the transcriptomics, you've got the metabolomics, you've got what? Phenomics, you know, everybody wants to do an omic now. So, but if you are good with data, then you have an edge. I don't know how long that will last, but if you're really interested in doing research, this is probably an area that you might want to consider. And it doesn't use a lot of money. You just need to use your computing skills and then data mining skills. All right. So paper number 29, tremendous advances, review of status and methods. All right. Nothing well, I wouldn't say nothing interesting, but nothing out of the ordinary. Anyway, this is something unique. Snitch. I don't know whether you learn about Switch in second year. Switch is an RNA that falls into a particular structure and it responds to proteins, it responds to metabolites and things like that. So this is a Switch. But what is a Snitch? Snitch is an element of RNA structure with a specific function that is disrupted by a single nucleotide variations. All right. So it says they're more concerned about structure. All right. So it's not so when you talk about sequences, the function one is based on its enzymatic function. The other one is structural function, how it folds, how two RNA molecules can maybe combine through interaction and things like that. So they're talking about function, and this group is interested in looking at function structure. Note, this paper uses single nucleotide variation, SNV. You know, why do you not use SNPs, SNPs? But by general acceptance, there's no rule. By general acceptance, you know, normally when we say mutations, we are normally referring to something that's negative, right? Mutation is just a process of change of sequence. It could be good, like X-Men. You know, there are a lot of mutants there, but they are actually superior. I mean, that is a comical example, but there are other mutations that are good. But for some reasons, when we talk about mutations and diseases, we're talking about variations that lead to something that's negative. And then you have mutations, and then you have polymorphism. Polymorphism is also a change, but we use the term polymorphism to signify that the change is not detrimental, is not problematic to the individual, right? This is by convention, by general acceptance. The whole worldwide, when we talk about mutations and polymorphisms, we know what it means. That's why this group says, you know, you cannot use SNPs, single nucleotide polymorphisms, because they want to show that the changes affect function. So that's why they said single nucleotide variants. I'm not sure whether that term is highly accepted or not. They mention about this ultra high throughput structure probing. So they look at the study and look at the structure of protein, RNA. Okay. Still no questions, right? Okay. Next, single nucleotide polymorphisms in clinics, fantasy or reality for cancer. So just a review on whether NGS is ready for practical use in the clinics. Can they really use this, like, oh, get some sample in and then do a genome study and then come up with all predictions? This is what I want you to have a look at, and you can have a look at it now. The websites, I'll just use here, right? The website name is 23andMe. The other one is Ancestry.com. Okay, I mean, these are the two, one of the first two popular companies that offer genetic screens. All right, have a look at them, because this is part of your, again, assignment for your exam evaluation. Okay. Okay, moving on. 2016, methods, challenges, and future development. Again, this one now talks about sampling sequencing for SNP genotyping. So it says, again, the issue is not being able to compare across different studies because of variation in methods. So, you know, so they propose we review major steps involved in library preparation, the type of adapters used for ligation, the primers designed for amplification of ligated products, yeah, things like that. Okay. This is what, number 33. Okay. Now, this paper is, I wouldn't say, well, it's actually quite ambitious. It says that why do you have a look? Why do you look at all these different mutations? Why don't you just look at selected variations, right? So it says reduce complexity. So it says reveal numerous genomic hits associated with complex phenotypes. In most cases, this hits along with surrogate genetic variation as measured by numerous SNPs that are linked in linkage disequilibrium are not encoding genes making assignment of functionality or causality intractable. So I'm just saying that this group is just saying that too much data not easy to understand. Why don't you just remove all the data, only look at what you understand? So it's not really. How do you know whether a paper is actually accepted or not? Well, given time, this is what, 2016, so it's about 5 years. You see that it's only been cited by 6 articles. Citation is an important measure of acceptance, right? So if this they propose and in 5 years they've only received 6 citations, probably they're not that probably is not that prominently agreed by many people who does this work, right? Okay. Yeah, bear in mind example. Okay, again, there's something wrong here. Bear in mind, wrong examples are also frequently cited. You know, when people talk about saying that, you know, something bad about you, you know, sometimes, you know, that you are used as an example of something that's not correct. So that can be also highly cited. So these are things that you want to bear in mind. If you see high citation, most of the time it's good, but sometimes, you know, it's also a reference of what not to do. Number 34, review of status. Genetic architecture of complex traits is even more complex than previously thought. Yeah, why I find this funny, because I was reading through it and it says something about methods of predicting future phenotypes, collectively known as genomic selection. I said, what? Why are we talking about selection? Then apparently this is a paper not on human diseases, but on agriculture. Complex and qualitative traits are important in medicine, agriculture, and evolution. But this paper specifically talks about complex traits in agriculture. That's why I talk about selection. We're not trying to be, you know, eugenics. Have you heard of the term eugenics? Where shall I write it? I shall write it here. E-U-G-N-I-C-S, eugenics. Eugenics is the practice of selecting superior genotypes against inferior. They say previously, historically, there was a group of scientists who believe that bad behavior is mostly genetics. So that means it says the criminals have a particular genetic makeup, thieves and robbers and rapists and murderers have all this particular genetic makeup. So they are selected against in the culture. So we only want to select only good genes that do not have that behavior. So this is, and then there was this elimination of these people. So that's eugenics. Very controversial. Okay. I mean, following from this, there's a lot of papers that are just updates and on methods. This is 2016. Again, updates on influence of genetics in sports. Now, the next paper is interesting. Well, in 2017, it says, are genetic polymorphisms in renin-angiotensin-aldosterone system associated with essential hypertension? Evidence from genome-wide association studies. Now, this study, why am I saying it's interesting? It says It says previously in the candidate gene era. What is candidate gene? That means you only study selected group gene. In the candidate gene era, dozens of SNPs with renin-angiotensin-aldosterone system (RAS) have been reported to be significantly associated with hypertension. That was a paper studies published previously. Now they looked at the same patients, and then they're not just looking at the RAS, but they look at genome-wide and said, after data processing, here it is, we found that polymorphisms REN, AGT, ACE2, CYP11B2, and so on were not associated with any disease trait. So earlier studies says there's association, now this says no. And then it says, additionally, SNPs with ACE, AGTR1, AGTR2, again, previously said important in hypertension, says it has associated with other traits, but not hypertension. Right? The only SNP associated with blood pressure trait was this. There's only one SNP. However, they don't know what gene is because it's actually an element. All right, and says therefore the effect of RAS polymorphisms may have been overestimated. Now again, this is RAS, not RAS gene for cancer. The effects of RAS polymorphisms may have been overestimated during the candidate gene era. See, so I mean, personally, at one point I was doubting all these NGS studies because it comes up with only statistics, maybes, right? So that's why I tend to focus really on the single gene disorders. Okay. Again, status studies. This is 2017. This is a study when we talk about genetic traits. Normally we refer to genetic diseases, but as shown here, infectious diseases also have genetic components, right? So this is paper number 38. Genetic factors that have been identified include genes encoding virus receptors, receptor modifying enzyme, and a wide variety of innate adaptive immunity related proteins. So genes that are involved with immune system is also, because you're talking about invasion of viruses, and then that normally induces your immune response. So people who get diseases are normally those who are not able to fight. It says there are some determinants where it could probably be associated with immune response that individuals lack. So it means to say that you need to understand how these viruses attack. Not because previously it says previously they just study the virus and how it functions. They don't really look at the host. But now the host is a human. So now they're saying that you must look at the host as well, and not like for example, I'm sure you read cases where patients, 100-year-old patients survive COVID, right? 80-year-old patients survive COVID. So who says old people are severely affected? Of course, most of them may not have it, but there are cases that that survive. Okay, so it'll be interesting to look at genetics if you can actually make some comparisons. Again, obesity in 2018. BMI, this is only MI. BMI, waist to hip ratio, and other apostates have been identified in more than 300 SNPs. Okay, gene by environment and lifestyle interaction. Right? Note here, always remember P is equals G plus E. You learned that somewhere, right? P is your phenotype, is in most cases it's the interaction between genetics and environment. Environment can be outside, can be what you eat, can be cellular environment, but you know what you see at the end, your phenotype is actually a combination of both. So genes by environment lifestyle interaction have revealed that our increasingly obesogenic environment, is saying that, you know, our obesogenic environment means the environment is actually leading towards obesity, you know, with all the eateries, the food, the cakes, the bubble teas and so on. You know, my daughter loves bubble teas. I try to drink them, they're just so sweet. So these kind of things, yeah, they actually, and with all the fast food restaurants that we have now, so this is what we call as the obesogenic environment, might be amplifying genetic risks for obesity. That means you have the risk, but living in this environment, that means you have a higher risk. But yet those at highest risk could mitigate. You can control it by increasing physical activity. So you have to exercise, which is not really helping because now you have all these gadgets, blah, 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 blah. And possibly avoiding specific dietary components. Eat healthy, right? Live healthy and eat healthy. There's a term called, I'm not sure whether I'm sure you have heard of this. There's a term called lifestyle diseases, right? So these are diseases of the lifestyle, the things you eat, the what. So they have an effect if they increase the effects of whatever risk you have. Okay, that's paper number 39. 40, pathway. Analysis of tools to visualize genetic variants. So I remember I mentioned it's not to visualize variants. So it says, you know, you need to see. These are all people who are frustrated with the fact that there's a lot of data, there's a lot of association, but what's next? You know, you can't really see. You can just see a lot of links. But whether or not they actually mean anything, it's not really clear. So again, this is the method paper, visualizing of data pathway analysis. The next one is also a method paper, understanding and functional understanding. So again, you know, it says enough is, there was one conference that I went, they said, you know, enough is enough of all this sequencing. Find out what it means. You know, because every time you sequence, you come up with multitude of data, but there's no point in having all this variation, but you don't really know what it means. Yes, you can see association. If you have this or a higher risk, but if you don't know which one is doing what, there's very little information. So this is 2018. It's about 10 years from the initial beginnings, right? 2007 and now 2018, about 10 to 11 years. You know, it says we have all the data, we can get a single molecule, we can get all the variants, but we need to know the function. Okay, functional ascending, and then you can have a treatment plan. Next paper, methods, also is a paper, this is what, paper 42. So this one, okay, the method 41 talks about understanding the disease, how you can use that to plan treatment. This paper, again, talks about understanding, but not to treat, but how to use it for prediction, right? Again, okay, 43, from identity to function, current strategies again. So I'll just read this bit. This is highlighted in green. It says, we are now confronted with a general lack of understanding how these, hang on, there's something on my, yeah. We are now confronted with the general lack of understanding of how these traits associated variants act. How do these genetic changes lead to overt clinical phenotype? Overt means clearly different. Yeah. What are the molecular mechanisms? Can we harness this information to develop better preventive and curative strategies? So this is again highlighting the problems, but not really providing any solution at the moment. 44, I mean, you'll see that papers, a lot of the papers in more recently are talking about the various challenges, the various ways of people doing and how to go about it and things like that. So brief overview of a decade of genome-wide association studies with primary hypertension. This one for hypertension is actually a Malaysian paper, I think. We have learned that GWAS about hypertension. Yes, a lot of association, but what does it tell you about the management? How do you manage the patients? 45. There's now a lot of potential to use all data, although there's still challenges, low sample size and incompatibility of results. All right. Okay. Previously, what paper number? This is 46. Previously, there was this paper about virus. So this one is also the same thing, but this one is more broad to infectious diseases. It says infectious diseases, much focus has been given to the pathogen. Right? Previously, we studied the virus, we studied the whatever. I mean, infectious diseases, you're talking about the virus, the bacteria, or whatever. And even in dengue, for example, we study the virus, we study the mosquito, the Aedes. Host genetics also contribute significantly to infectious disease burden. So this is 2018. So it says no, some people can and some people can withstand it, but others don't. So you need to find that out. This paper is like the virus infection paper, but more general to infectious diseases. First, we introduce observational twin-twin concordance. Concordant discordance, here you study twins and then say, we study twins because they had if one is exposed to this kind of environment, what will be the other? So this is the twin-twin concordance studies. Diseases of such as poliomyelitis, tuberculosis, hepatitis, which suggest the important role of host genetics. We review the well-established links between specific genetic alterations and predisposition. Predisposition is like a high risk of getting malaria. And we discussed the novel findings yielded by modern GWAS studies, which suggest strong contribution of immunologic variations in your immune system genes in the MHC. These are your genes that makes your antibody molecules to host genetic infectious disease susceptibility. All right. So yeah. And paper number 47. You got 3 more papers. 47 says, yeah, 2019. People are still talking about how best to study. All right, understanding non-Mendelian, because that's what the situation is now. You have a lot of data. There's a lot of sequence variation. There's a database here set up and so on. But what does it all mean? So that is the crucial missing answer at the moment. So people are still talking about how best to do study, need to understand what variations do. And finally, paper number 48, still the problem of not understanding what variance means. It says genome-wide association studies have successfully mapped thousands of loci associated with complex traits. These associations could reveal the molecular mechanisms altered in common complexes in result of identification of novel drug targets. So if you can understand that, you can plan the treatment. However, GWAS has also left a number of outstanding questions. For example, the majority of diseases associated lie on non-coding regions of the genome. So even though they are thought to play a role in gene expression, it is unclear which genes they regulate. It's not just about how they function. They don't even know what gene is affected by that particular variation. Yeah, it is unclear which genes they regulate and which cell type of physiological context this regulation occurs. This has hindered the translation of GWAS findings into clinical intervention. So that means if you wanted to use this for treatment, you have to understand the disease. All right. So, so that sort of sums up the NGS and GWAS. So I hope you are able to synthesize some information from 2007 until about now 2020. That is almost 2000, yeah, about 13 years, if my maths is correct, 14 years. And you see the development of thinking, you know, from getting data, get data, and then say, okay, finding association with data, and then okay, now you find an association, but you are left with this association. So what's next? Because in order for it to be useful, you need to know what it does. Then only you can plan treatment. So then a lot of talk now is saying, okay, how do you find association? How do you make comparisons? How to sieve all the data and see what matters. So there's a lot of data that people think is really trying to decode, you know, genetic information and try to understand how these things work. It's not easy because, yeah, looking at coding sequences, that means you can identify, you can really say, oh, this mutation changes the protein amino acid, but if it's on a non-coding sequence, you know, then you don't know whether it changes the structure, whether it involves in binding, whether it involves in expression, so you don't know, and you don't even know which gene that is involved. Now, this is the hardest part at the moment. But having said that, this opens up a whole new potential area of research in bioinformatics and data mining. Right? So if you're really interested, I would actually advise you to go into that. Because once you have that capability, then it's really about looking at all data and playing around with and trying to find correlations. Okay. Hopefully I have done justice to tell you the different types of NGS platforms and what they can do and what people expect from it, and hopefully you've got some knowledge. And that was the intention. You need to look at, you need to be able to understand the powers of genomic studies and the challenges that it represents before you agree on doing something that probably get you in trouble later. All right. Okay. So any questions?